Hello, I am Dr. Doe, and today I'm going to be showing you how to solve DNA replication, transcription, and translation problems. So let's go ahead and get started. So here are the learning objectives. If you're given a DNA strand, you'll be able to transcribe that into RNA. And then once you have that RNA, you'll be able to translate that into a polypeptide/protein sequence. Now, in this video, I'm going to assume that you already have the basics of transcription and translation. You already know the details, what enzymes are involved. So I'm going to cut straight to the chase and show you how to do those problems. But before that, just to kind of recap, okay, I know we're just going to recap just a little bit here. Very quickly, you do start off, all your genes are found in your DNA. When it comes time to pull out one of those genes to produce a protein, you get RNA. Sometimes professors will write mRNA, same thing. And then finally you have a protein. Okay. Now when DNA tries to make a copy of itself, this is called DNA replication. When DNA tries to make mRNA, actually when DNA is used as a template to synthesize RNA, it's called transcription. Okay. And then when mRNA is used as the blueprint to synthesize protein or a polypeptide protein, now we have that process is called translation. And again, amino acids are the building blocks for these proteins, for this polypeptide chain. Okay, let's move. Now the DNA has 4 letters or 4 mononucleotides that you will find across in that DNA molecule. And the first of those letters is A, another one is T, G, and C. Now say for example that we are replicating DNA. So for every letter A that DNA polymerase enzyme sees, it will lay down the letter T. For every letter T, it's going to complement with the letter A. For every letter G, you'll get the letter C. And for every letter C, you will get G. That's the rule. Now, if you have DNA and you are now trying to synthesize RNA, then for every letter T that the RNA polymerase sees, it's going to put down the letter A. That's its complement. For every letter C that it finds, it's going to put down the letter G. And for every letter G that it finds, it's going to put the letter C. Now, I skipped this one for a reason because a lot of students think that for every letter A that it finds, the RNA polymerase is going to lay down a different letter. A lot of students think it's going to lay down the letter T, but in fact it doesn't. It lays down a different letter, and that letter here is the letter U. Very important that you know that, okay? And that's different because the letter A, if you have DNA to DNA, for every letter A that it sees, we'll get T. But then when it goes from DNA to RNA, for every letter A that it sees, we get here the letter U. And you're going to need to know these rules. Now, if you have the RNA and you're trying to make a protein, there is no such thing as letter G's going there, making some letter C's. None of that exists, actually. But in order to do so, you need to use something called the genetic codon or the genetic code chart. And I will show you guys that in a few minutes. Actually, I'll show you guys that right now. So here's a slide showing you the genetic codon chart, okay? And what happens here is that the ribosome, which is the machine, the organelle machine that is used, what happens at the mRNA, the ribosome docks onto the mRNA and it starts to scan across that mRNA and it reads each of the RNA alphabet letters. Remember in the last slide, the alphabet letters in DNA are A, T, G, and C, but the alphabet letters for RNA are A, U, G, and C. Okay? So in this string of RNA, The ribosome is going to read this huge string of RNA in groups of 3 letters. But in order for the ribosome to know where to start, it's going to first scan that RNA strand, and it's going to look for this major codon. It has to tell that ribosome this is where you have to start, and that here is AUG. It must find that AUG before it can start working and doing its job of translating and making polypeptide chains. Okay, so the AUG is extremely important. All right. Okay, so for example, AUG, here's how you would use this genetic codon chart. AUG is the start codon. It's got to find that. So if you have those 3 letters, here's the letter A. Okay, if I want to know what amino acid goes there, I can use this chart by the first letter here is A. Look for the letter A. That's the first letter. Great, I circled it. Second letter is U. Well, the second letter is on the top, so here's U. So I find where the column and the rows meet, and it's in this box right here. We already know the answer, right? It's already in this box. And then now that last letter is a G. So AAU, nope. AUC, nope. Ah, here's AUG, and we have this MET. That's the 3-letter code. Okay, so on my other chart here, I have MET, and that encodes for methionine. That's the name of this amino acid. Here's one more example that I'm going to show you. What if I said AGU? How about AGU? Well, the first letter here is A. First of all, let me kind of get my little eraser out here. I want to erase this so we can redo this. Okay, let's get my pen. Okay, so the first letter here is A. Let's find A. So, here's A. Second letter here is G. Here's the second letter. That should say second, not Sion. G. Where does the row and the column meet? Right here in this box. Oops, that was pretty bad. Okay, right here in this box. Did it again. Whatever, you get it. You get the idea. Okay, this tablet pen is a little tricky. And then the last letter is U, so A-G-U. We're lucky today because it's the first one, and that's S-E-R. And on my chart here, S-E-R stands for serine. Okay, so that would be the amino acid. That's how you read this genetic codon chart. Now, if you don't know how to use the columns and the sides here, if you can't remember on an exam how to read this, you're just going to have to read each one of these 3 letters one by one until you get the 3 letters you're looking for. Okay? All right. Let's get straight to demo problem number 1. I recommend that you pause this video and try it out. Otherwise, let's watch and see how I do this. Okay, always, when working with these problems, you always want to work with the template strand. Always, 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 always work with the template strand. These rules that we talked about over here only work with the template strand. Only the template. Okay? Only the template strand. Extremely important. Only use the template strand. All right. In our example, we're given that template strand. Very easy. All right. So we have DNA, and we're going back. We're trying to make a complementary copy of another DNA strand. Okay. So we have the letter A according to our rules. A pairs with T. Next, T here is A, then T, then G, C, T, right? Next one here is A, G, A, C, G for that letter here. T, G, T, T, C, T, A, G, G, A. Hope you got that right. Next up here, let's get the mRNA. So when you go from DNA to mRNA, the alphabet changes a little bit. Remember here that for every letter A that we get, it becomes the letter U, not the letter T anymore. Okay, so let's do this. All right, the DNA template strand here says A, so it becomes U, right? Put a little star there to remind you it's U. The next letter here is T, So that's A. Next one here is the letter T, so that's A. And then the next one here is letter A, that becomes U. And then we have here G, then C, U, A, G, A, C, G, U, G, U, U, C, U, A, G, G, and A. I hope you got that right. All right. I'm going to change my pen color here just a little bit. Uh-oh. That's not what I wanted. Okay. That's fine. Okay, so here's the thing. In order for us to work with this, remember I told you guys earlier that the ribosome has to find the start codon. And we remember that the start codon was AUG. So what happens is the ribosome is going to start here on the left side, and it's going to scan for the first three letters AUG so that the ribosome knows where to start. All right, so here we go. The first three letters are UAA. That's not AUG. Keep going. Next three. AAU. Nope. Next three. AUG. This is it. Okay, this is here. This is AUG right there. Okay, AUG. Now that doesn't look very nice. All right. We found our first three letters AUG, and I'm going to use my pen and I'm going to circle the first three letters AUG, and then the next three letters, and then the next three letters. This is a very, very important step, that you circle every three letters after that AUG until the very end. Okay, until you have no more letters to circle. Okay. Now I want the protein, the amino acid sequence, and I want the full name. So the first three letters here are AUG. We already know that. It's methionine. Okay. The next one here is CUA. So let's kind of go back. Where is CUA? Well, here's C, here's U, CUA is LEU. According to my list here, LEU is leucine. Okay, let's write that down. Okay. Okay. Next one here is GAC. Well, what's that? Let's take a look. G is right here. A is up here. So where the row and the column meet is in this box. And GA, I believe it's GAC. GAC here is ASP. And in my chart here, ASP stands for aspartic acid. All right, let's write that down. Okay, I ran out of room. I'm just going to move on to the next one. All right, this GUC here, that's the next GUG. What's GUG? G, U, and G. Here it is. That's val. But what's val? In my chart, VAL is down here at the bottom. That's valine. All right, let's write that down. Next one here is UUC. Well, let's see what that is. Here's U. Here's U. And C is, here it is. UUC is PHE, phe. So what's that? PHE, here it is. Phenylalanine. Let's write that down. Phenylalanine, that's my E. And then the last one here is UAG. Well, what's that? U is here, A is here, and UAG. That means stop. Uh-oh. What does stop mean? For me, stop means don't write anymore. Stop writing. But a lot of bio professors, what they're going to do is they'll say, okay, just write the word stop. So you have to check in with your bio professor. Okay, but if you're in my class, usually I will say just don't even write anymore. Why? Because the word stop is not an amino acid, and that's why usually I omit that. Okay, so here's the answer to demo problem number 1. Hope you got that right. All right, let's go ahead and move to demo problem number 2. In this example, you're given something called the DNA coding strand. That's the opposite strand. So let's go ahead and work on this problem. We are in a little pickle though, because remember I told you always use the DNA template strand. You have to find this. All right, so let's go ahead and do that. We know that if you have the DNA coding, if you take the complement, you'll eventually get to the template. So for every letter A, we have the letter T. Next letter here should be A. Then here is A. For the A, it becomes a T. And then we keep going. Keep going on this. Here we go. C, G, T, G, T, T, C, T, A, T, C, A. There's our DNA template strand. Okay. Now I need to find what the mRNA strand is. Okay, remember for every letter A, it doesn't become a T anymore. Now it becomes a letter U because the alphabet letters are a little bit different for mRNA. So let's do this. Template strand here says the letter T, so mRNA should be A. For every letter A, we get the letter U. A becomes a U. And then for T becomes an A. This one here we have the letter U, then G, then A, then U, C, U, G, C, A, C, A, G, A, U, A, G, and U. Did you get that answer? I hope so. All right. Now we need the protein amino acid sequence. We're going to use a 3-letter code, but before we can even find that, we need to know where the ribosome is supposed to start. So I'm going to go ahead and change the color of my ink here. Blue color. I need to scan every 3 letters until I see the first AUG, starting with the left, right over here. AUU. Nope. Next one. UUA. Nope. Next one. UAU. Nope. Next one. AU. Ah, here it is. And there's our first AUG. And I'm going to circle every 3 after that until I get to the very end of my problem. There is the next 3 and so on. There are no more letters left to circle. All right. So now in this protein sequence, it wants me to use a 3-letter code this time. All right. So AUG, what's that? According to our chart here, AUG is methionine. So it's MET. Let's write MET down. Again, we are using the 3-letter code. Dash, what's AUC? Here's A, U, and C. It's ILE. Okay. Let's write that down. ILE. Now we have UGC. What's that? Here's U. The second letter is G. UGC is here. It's CYS. That's cysteine. CYS. Let's write that down. Next one here is ACA. What's that one? Here's A. C is here. And ACA is THR. That would be threonine. So let's write THR. And then AGA, what's this? Go back to my chart. Here's A. Here's G. And AGA here is ARG. What's ARG? It's arginine. Okay, let's write that down. And then lastly is UAG. What's that? U is here. Here's the first letter U. A is here. UAG. That's a stop codon. Okay, so we don't write anything technically, but just for completeness, we'll go ahead and write stop, but you know you're not supposed to write that. Okay? This is our 3-letter amino acid sequence. Hope you got that right. All right, here's the third demo problem that I'm going to do. We're given a DNA template strand. Sounds pretty straightforward and it's very similar to the first, so let's do it. Okay, for every letter A becomes a letter T in the coding strand. DNA, remember DNA to DNA, so we can use the letter T. And then T here becomes an A, T here becomes an A, and then A becomes a T. Then we have a G, C, T, A, G, A, C, G, T, G, T, T, C, T, A, G, G, A. All right. Looks about right. All right, now we need to go to the mRNA strand. Remember, the alphabet letters are a little bit different. So for every letter A on the template strand, it becomes a letter U. Then we have here A, then A, U, G, C, U, A, G, A, then C, G, U, G, U, U, C, U, A, G, G, A. Okay. Great. Almost done. All right. Now from RNA to protein, don't forget we need to find the 3-letter codon that stands for start, and we already know that's AUG. So let's go ahead and find that AUG. The first 3 letters here are UAA. That doesn't mean start. Next one, AAU. Nope, that's not a start. How about the next third one, AUG? Ah, we found it. Here's AUG. Okay, remember, starting from the leftmost side, and then circle every 3 letters after that. Here we go. Every 3 letters after that. No more, no less. Every 3 letters after that. All right. Now, what is the amino acid sequence? And in this problem, it wants me to use the 1 letter. Okay? Okay, so here we go. AUG, we already know that's methionine. Okay. Oops, what the heck happened to my slide? All right, we already know. So AUG here stands for methionine, and the 1 letter is the letter M. So let's write that down. The next one here is CUA. What's that? CUA, here's C, here's the letter U. CUA is LEU, L-E-U. So the 1 letter is the letter L. Let's write that down. The next one is GAC. Let's take a look. GAC, where's that? G, A, and C is ASP. ASP, the letter here is D. All right. Let's put the letter D down. Next letter here. Okay, so GUG. Let's go back to my chart. Here's G. Here's U, and here's the letter G. The vowel. So the letter, 1 letter is V. UUC here. Let's see. What's UUC? U and C is PHE. That stands for phenylalanine, and that letter here is F. According to our chart, it's F. Let's put that down. The last one here is UAG. What's that? Let's see here. U, A, and G. That means stop. I'm just going to go ahead and write stop for completeness, but you're technically not supposed to write that. All right. So that is our demo for number 3. I hope that you found these 3 problems helpful, and I hope it's trained you well in solving any of these types of problems. I have 3 more problems that you can do on your own. Please try them out. And pause the video right now, because in a few seconds I'm going to flip to the slide that has the answer. So go ahead and pause the video and practice on this first, and then play the video again and I'll show you guys the answers. All right, pause. Hopefully you paused, right? Okay, so here are the answers. I hope you got that right. Thank you very much for watching. I hope that you found this informative and helpful, and go ace those problems on the test, okay? From here in Southern California, sayonara. I'll see you next time in the next video.